Hi everybody, this is Ms. Peachy from WCA Biology, and I'm doing a quick review here for your cells unit test since I won't be in class to do a review on Thursday. So I'm on my website currently. You guys can access this if you don't know how from the pushpin icon at the top of your screen. And I clicked on the biology tab at the top of my website to get to this page. Remember, when you do take your unit test on cells, you'll be clicking on this link, Unit 3 Test Cells. And for the review, we're going to be using the study guide for cells test. When I open that up, I come to this page here. And this is not something you can edit because it is a master copy. So in order to edit this, you're going to either have to file download to Microsoft Word, download it for yourself, and you can save it and edit it on your computer, or you can hit make a copy, which allows you to use Google Docs to edit it right online, and it will automatically save that for you. So that is what I did. And the first thing you'll see on here is we do have all of the essay questions are here for you. And I'm going to kind of walk you through what I want for those essay questions, but I'm going to leave up some of the research and things like that for you. I will kind of direct you into where to go to find the information. But remember that for these essay questions, you are allowed, since I give these to you in advance, to prepare your essay questions in advance, research them, write them all up. Make sure you're using just your textbook or this review session or your lessons or any of my recorded classes and stuff like that as your resources, though I don't want you to be Googling answers for these questions, okay? So for the first question says use the organelles to follow the path of a protein being made. Make sure to include not only where the protein goes, but what happens in each location. So just like the organs in your body, you can follow the blood as it moves throughout the cardiovascular system in your body as it leaves the heart, and through the different vessels to the lungs and back to the heart and through different vessels. It has more than one organ that it interacts with as it kind of moves throughout your body. So does a protein then interact with more than one of your organelles in a cell as it leaves the cell. To kind of best summarize what's happening, I do want you to watch a short video clip on this one. So I'm going to embed that right into this video.
So what are organelles? Organelle means little organ. Organelles are the specialized parts of a cell that have unique jobs to perform. Let's start with the nucleus, the control center of the cell. The nucleus contains DNA, or genetic material. DNA dictates what the cell is going to do and how it's going to do it. Chromatin is the tangled, spread out form of DNA found inside the nuclear membrane. When a cell is ready to divide, DNA condenses into structures known as chromosomes. The nucleus also contains a nucleolus, which is a structure where ribosomes are made. After ribosomes leave the nucleus, they will have the important job of synthesizing or making proteins. Outside the nucleus, the ribosomes and the rest of the organelles float around in cytoplasm, which is the jelly-like substance. Ribosomes may wander freely within the cytoplasm or attach to the endoplasmic reticulum, sometimes abbreviated as ER. There are two types of ER. Rough ER has ribosomes attached to it, and smooth ER doesn't have ribosomes attached to it. The endoplasmic reticulum is a membrane-enclosed passageway for transporting materials, such as the proteins synthesized by ribosomes. Proteins and other materials emerge from the endoplasmic reticulum in small vesicles, where the Golgi apparatus, sometimes called the Golgi body, receives them. As proteins move through the Golgi body, They're customized into forms that the cell can use. The Golgi body does this by folding the proteins into usable shapes, or adding other materials onto them, such as lipids or carbohydrates.
All right, and once the Golgi body or the Golgi apparatus modifies those proteins, they actually ship them out in more vesicles. And if the proteins are meant to leave the cell, the vesicles then will go to the cell membrane and they will be exported out of the cell. And that process is through something called exocytosis. So they actually go to the cell membrane, and the cell membrane attaches to the vesicle and kind of pulls that whole vesicle out of the cell to allow that protein to leave the cell to be transported through the bloodstream someplace else in the body where that protein might be used. So I want you to make sure you can use those terms. There's more than what is on this list in that video clip, but those include, the nucleus, ribosome, endoplasmic reticulum, the vesicle, the Golgi apparatus, and the cell membrane. So put them in order and discuss what's happening in each location. Question number 2 asks about using the concept of osmosis and either hypertonic solutions to kind of explain why freshwater fish would die if placed in saltwater, and saltwater fish would die if placed in freshwater. For question number 2, I would like you to look at page 210 in your textbook. Okay, so I want you, 210 and 211, I want you to understand what the difference between a hypertonic and a hypotonic solution is, and remembering that a saltwater fish is adapted to life in saltwater. So that means its blood is going to have a higher concentration of saltwater, or salty ions in it, compared to if it were placed in fresh water, it would have a higher concentration of salt inside the fish than inside the water. If a freshwater fish is adapted to living in fresh water, it would have a lower concentration of salt inside of the fish than the salt water you would place it into. So keep that in mind when you're discussing the answer to question number 2. Looking at pages 210 and 211. Okay, for the third essay question, you need to think back to the portfolio that you did on cell size using the potatoes. So for this one, you need to explain why it's more advantageous for a cell to have smaller cells instead of larger ones. And I want you to take into consideration comparing the ratio of surface area to volume for this one. So this is going back to Unit 3, Lesson 4. Okay, and this is the lab that you did, the portfolio, talking about why, basically why is it an advantage for a cell to have a smaller sizes in cells. Okay, so go back to this lesson, review what we said in the lab, and then that is what you're going to report on for this particular question. Keep in mind, I know some of your labs didn't exactly work out the way they should have, but think about it this way. If you're a large cell, you need a lot of cell to be able to get stuff to all parts of the cell, right? So when you, as a cell, transport materials into the cell through diffusion and osmosis, it's much more difficult to be able to get enough stuff into all parts of the cell if the cell is bigger. Not only that, but the cell has more waste products. So transporting waste out of the cell also becomes equally more difficult. You can kind of compare a cell to maybe a country or a large family. Is it easier to feed a small family, or is it easier to feed a large family? And if you're a family and you have a couple people living in the house, is it easier to keep that house clean if there's a smaller family? Or is it going to be easier if you've got, a lot more space to clean and a lot more people living in the family, right? So you can kind of use that as an analogy to help you kind of think about why small cells are more advantageous, excuse me. And then finally, with question number 4, I want you to explain what happens during the interphase portion of the cell cycle. Okay, so for this question, you're going to go to your biology. This is on lesson 15. So in lesson 15 on slide 3, there's actually a link that asks you this very question. It says what happens during interphase, the G1, the S, and the G2 phase. And if you click on this link, it actually is going to outline right in here what's happening during this part of the cell cycle during interphase. So again, reread this, put it into your own words. Don't just, write what's on here. But that's what you're paraphrasing as part of that answer for the test. Okay, let's go back to your study guide real quick here. And just a few other parts that you need to know. It says the difference between a solute, a solvent, and a solution. So please know that, right? That a solute is what you dissolve into a solvent. When you mix a solute and a solvent together, it becomes a solution. Know what osmosis is. Know when you comes to diffusion, which direction the solute's going to move. Is it going from high to low concentration or low to high concentration? Again, we talked about this for one of your essay questions, but be able to label solutions as hypertonic, hypotonic, or isotonic, and tell me if the cell is going to shrink or going to expand. And that's in your textbook as well. Back in, let's see, it's back in chapter 7, section 3. So 7.3 on my book is pages 208 to 211. So it's going to talk about those different types of solutions and whether the cell will shrink or expand. Give the order of events in cellular respiration. The 4 things you're going to be looking for, guys, are like glycolysis, you're looking at pyruvic oxidation, the Krebs cycle, and the electron transport chain. Where does glycolysis take place? Look that up. Where does the Krebs cycle take place? These are specific parts, so make sure you're very, very specific about where they're taking place. What is ATP? Where do the light-dependent reactions take place in the chloroplast? Remember, the 2 parts of the chloroplast are the stroma and the thylakoids. So which part is the light-dependent reactions and which part are the Calvin cycle? Understand what happens to water in the light-dependent reactions of photosynthesis and what 2 energy molecules are made in this part of the photosynthesis. Remember that light comes in and actually strikes water. And that water is split apart. Oxygen gets released. The hydrogen actually gets shuttled into the interior of the thylakoid membrane. And electrons are then carried down the photosynthetic electron transport chain to produce NADPH. And then that hydrogen ion gradient helps to produce ATP. So the 2 energy carrying molecules that are made in that part of photosynthesis are NADPH and ATP, and that all starts with the splitting of that first water molecule. In aerobic respiration, you get most of the ATP made in the electron transport chain. Talk about mitosis, the different phases, prophase, metaphase, anaphase, telophase. Make sure you know what's happening in each of those phases of mitosis, kind of the main events, right? So the main events here in prophase, you've got your DNA, which kind of shortens and thickens into chromosomes at that point that you can see. You've got the nuclear membrane dissolving. Now in metaphase, they all kind of get attached to those spindle fibers and line up at the middle point of the cell. In anaphase, they start migrating to either end of the cell. In telophase, a new nuclear membrane forms in each of the 2 new cells, and then the end of telophase is actually cytokinesis, when the cell pinches up into 2 different new cells. But that mitosis, that part of mitosis is actually the going to be the division of the nucleus of the cell. So mitosis is nuclear division. It's where the nucleus divides into two new nuclei. It's part of the whole cell cycle. You can think of it as cellular division too if you want to get more simple, okay? So that kind of gives you the gist of what you need to know for this test. Hopefully that helps a little bit with your review, guys. If you have any questions or concerns, feel free to call me up or let me know. And good luck on your Unit 3 test.